Thank you very much, Taddy. Can everybody hear me okay? Good morning. It's a great pleasure to be back, at least for a day. And thank you, Taddy, for the invitation. And it's great to see some familiar faces. And some of you have a little more gray hair. I have a little less hair, but I think that's unavoidable. This talk will be somewhat informal, and probably some of these thoughts that I'll transmit this morning are familiar to you, being students of epidemiology and probably some history of medicine and clinical trials. But I wanted to sketch a little bit of the early historic thinking, elements of thoughts, regarding trials and where some of these ideas came from. And I hope you find this at least visually entertaining. So the first point is that there's a number of historical threads that one might pick up in order to see the origin of some of the elements that we treasure so much today in our designed experiments. And they're listed here, but I really am only going to discuss two of these. First and foremost is the concept of what it means to have a fair comparison in evaluation of medical therapies. And then secondly, which is hard to separate from the concept of fair comparison, is this conversation between rationalism, or originally dogmatism, and empiricism throughout the history of medicine, in fact, but particularly dealing with experimentation. And these insights that we'll see with regard to both of these themes, the other three, unfortunately, I'll have to neglect today because of time. But these themes always come packaged in a somewhat interesting way when you look at the historical figures who embodied them. They come packaged in a way where there's a flash of insight from somebody, but simultaneously that persona or that scientist or that event in some cases also contains something that is either contrary or in some cases really surprisingly different or surprisingly the opposite. And I thought a good metaphor for this might be this Hieronymus Bosch painting here titled, it's from 1490, it's titled Cutting the Stone, or the Cure of Folly. And you can see in the chair is a Dutch fool figure who is having the flower of folly cut from his head. And in symbolism that is somewhat lost today, you can see the church looking on. There's probably mania escaping from his head in the thinking of the time. The woman is a representative of melancholia, the one with the book on her head, and that's a symbol of the time for false learning. And there's also other symbols here that don't translate well. And actually, the first time I used this picture, a very learned fellow at Medical University of South Carolina took the trouble to send me a letter, which I won't read to you, but he details all of the symbolism in the picture. It's really quite rich and not unlike some of the history of clinical trials. And I'm sure some of you probably remember a student here, Kara Weiserbs. Kurt, am I saying that name right? I think she was an art history major, and she saw this picture and also had some useful insights regarding this and the connection to clinical trials. So this notion that people are doing things, that folly is escaping, that there's this mix of symbolism and pressures on trials, I think you'll see in some of the topics that I'm going to cover. So here they are, assuming I get through them, and I will stop with about 15 minutes left, Taddy, so that if there are questions or discussion, and my guess is I won't make it all through all of these. Fair tests and why they're needed, the principles of testing, the role of chance, comparisons and biases, masking, and so on. Recent developments, I think, will probably be out of reach for the amount of time. Here are the take-home messages. I think one is that there's a dialectic between rationalism and empiricism over the centuries, and that only the proper balance of these two results in this entity, this mix of science that we call clinical trials. And I don't think that we could have trials in their present form until science had come to terms with this appropriate balance. And as I've said, the insights that I'm going to discuss are always packaged in a very imperfect but interesting way. So to be sure we're talking about the same thing, the sine qua non of a clinical trial is that the investigator controls the treatment administration. I think that's it, personally, in terms of definition. But there are other loci of control that we typically try to use. We try to control random variability and error. We try to control systematic error and extraneous effects. But these are not, in my opinion, absolute requirements of the definition. There are always comparators in a trial. In many cases, for prototypical trials like randomized studies, those comparators are internal and explicit to the experiment. But in other cases, they're external and only implicit, but they're always there. And what we always try to do is to make all things equal, everything except for the controlled treatment administration. If we look at the timeline from before the current era, hundreds of years, this is what it looks like early on. I'll mention a few of these names. One of the starting points often taken is an excerpt from the book of Daniel in the Bible. I'm not sure that's legitimate, as I'll mention, because I think many of the background concepts weren't present, but it is an interesting snippet. Celsus, in the lower middle there, was a 2nd century Greek philosopher who was in opposition to Christianity, and he had a lot to do with the empirical method, as did Philinus of Kos, the same birthplace as Hippocrates, and he's generally credited with being the founder of the empiric school. If you look outside the sort of Western tradition into Arabic medicine and also Asian cultures, you find some pretty interesting events and repositories, especially during the Dark Ages. And one in particular is from Ben Cao Tu Jing of the Song dynasty. This was in the year 960 to 1279. And he produced a work called the Atlas of Materia Medica. And an interesting quote is said that it was said that in order to evaluate the effect of genuine ginseng, 2 persons were asked to run together. One was given the ginseng while the other ran without. After running for approximately 3 to 5 li, but that's equivalent to about 1,500 to 2,500 meters, the one without the ginseng developed severe shortness of breath, while the one who took the ginseng breathed evenly and smoothly. So even in that little mini experiment, there's some pretty sophisticated concepts. But again, these ideas crop up and then would be lost for a time. If we move forward a little bit, we can see maybe some more familiar names. I'll touch particularly on Paracelsus as one of the literal embodiments of empiricism in medicine. Francis Bacon served as both Attorney General and Lord Chancellor of England, and he was influential even though his political career ended somewhat in disgrace. Roger Bacon was the English philosopher, and he is often credited with laying out the foundations of the modern scientific method. Interestingly, Thomas Percival, and I'll quote a bit of his work in a few minutes, was an English physician and author. He originated the first code of medical ethics, and it actually coined the term medical ethics. If we move forward to more familiar turf in the last or almost current century, there's some very important landmarks here as science matured, and we can actually point back, for example, to the use of randomization, the Santa Chryson trial, which in some people's book was considered to be the first randomized trial. This history is somewhat more familiar, I think, to this audience, and I probably won't touch on this very much. So let me discuss FAIR tests and why they're needed, then back to my outline. And here's part of the reason that we need FAIR tests. And the picture in the lower right should be familiar to you because it was here on this campus. And it represents a kind of authoritarianism. You could argue that these were the founders of this institution. This dates from about 1905. William Osler, Howard Atwood Kelly, William Stewart Halstead, and William Henry Welch. And this kind of power and authoritarianism, which in some sense was the residue of earlier authoritarian errors, is part of the reason that we need fair comparisons, because we can't take forward therapeutic opinion and knowledge without FAIR tests. In the upper left, you can see one of many, many such historical figures. This one comes from the dome of a chapel in Italy, and you can see a conversation there between Galen and Hippocrates. And again, it exudes the authoritarianism of what had been taught historically for hundreds of years. Never mind that Galen and Hippocrates were not contemporaries and could not possibly have sat literally and talked to one another. But the notion that they were somehow in conversation through the dogma of the times is an example of why we need these FAIR tests. If we take a very modern perspective on this, we need to give patients the most favorable risk benefits. We all know that. And prevent misleading claims, minimize errors, advance knowledge, use our resources appropriately, and recognize that FAIR tests are often directly in opposition to the kind of authoritarianism. Here's an example, and I'll also touch on this in my talk this afternoon. Many of you are too young, but some of you are not, to remember Benjamin Spock, who was a very influential child health expert, and he advised it through these writings, which 35 cents, for those of you who don't know, was an appropriate price in the 60s. He advised that babies should sleep on their tummies in case they should vomit, and that would prevent aspiration. And according to Gilbert and colleagues who reviewed this evidence, they think that this advice may have contributed to the death of tens of thousands of babies through crib deaths. It seemed very rational at the time, but the advice was not founded on any evidence whatsoever. So this is a very poignant example of why we need fair comparisons. Here's another example from, and as a general rule, I'm not going to respect calendar time. I will bounce around throughout history. Van Helmont in 1648, this is a quote from his opus on medicine. And listen to the mixture here of sophistication and political incorrectness. Let us take from the itinerant hospitals or from the camps or elsewhere 200 or 500 poor people with fevers, pleurisy, and so on. Cast lots so that half of them fall to one treatment and the other half to the other. I shall cure them without bloodletting or purging, and so on. So here's a direct challenge to one of the most cherished treatments of the time, bloodletting. And a sophisticated notion that we take people and divide them by randomization, if you will. Yet the notion that we're going to do this with poor people, of course, doesn't play. But otherwise, it's a rather sophisticated thought. So let me skip up then to comparisons of experience. Now, as epidemiologists, of course, many of you are very used to thinking and taking advantage of experience bases that maybe weren't constructed explicitly in the style of the definition I gave earlier for a clinical trial. But definitely through the evaluation of experience, we look and see biases. We can account, at least in part, for the effects of chance. We may actually design the data production for appropriate purposes, and we can address genuine uncertainties rather than trivial ones. So here's where we come, for example, to the book of Daniel. This is probably familiar to you about this dietary test. The end of 10 days, a direct comparison of who looked better, and it was clear that the children who ate the king's meat looked worse than those who had eaten pulse, which was a vegetable product. But at this time, as I hinted earlier, there wasn't real grounding in either science or medicine or critical reasoning of any kind, for example. So while this is, I think, an important notion, I'm not sure that it falls really in the direct line, at least not the one that I'm trying to draw with regard to experimentation. Comparison of experience was also written about in the Arab literature. And if we look, for example, at this document, Abd al-Latif ibn Yusuf, who would become known as Avicenna, repository of much medical wisdom during the Dark Ages. Pretty sophisticated idea here. I liked this because of the comparison to archery. The intelligent physician only errs occasionally, but will get things right 100 times. So it's an acknowledgement of error, but the skill makes these errors, if you will, tightly clustered around what should be true. Compare him to an expert in archery who mostly hits the mark, and when he misses it, it will not be far off, but land near the target. Now, this may be a little too glowing, but in a later passage, he says, and I liked this also because it was somewhat mathematical, the surface of the circle or square root. So here there was a fair amount of sophistication about what was calculable, if you will, rationally in mathematics. Someone skilled in this art determines this irrational number as closely as possible and will tolerate only a small error, the difference of which is not apparent. And so what he's saying is that errors are okay, but we only tolerate relatively small errors. Unsophisticated person will accept and make larger errors. So fairly sophisticated by our standards. I'm going to skip that next slide because the document there that for years I thought was supporting the point I wanted to make, it turned out when I called the Library of Historical Documents at Princeton University, where that document is held, it turned out it was not what I thought it was, so I'm going to skip that. Let me discuss some interesting history in the British Navy. It seems like an unlikely place for much sophistication about clinical trials to come forward, but I'm going to discuss this well-known history with James Lancaster and also the person who followed him, Lind. Interestingly, in 1600, Lancaster had a ship supplied with lemon juice, and 3 others in this fleet weren't so supplied. And Lancaster's men remained free of scurvy, and the correct insight was made. The reason why was he brought to sea with him certain bottles of the juice of lemons, which he gave to each one as long as it would last. And so the direct connection between the use of lemon juice and prevention of scurvy was known as early as 1600. If we look at the subsequent history, and incidentally, these documents show a very careful accounting of the reasons for incapacity in the British Navy. It's not necessary to read them, but on the right-hand side, you can see a long list of things with counts of the number of sailors incapacitated from things like gout, leprosy, lumbago, and so on. So they were very careful in their observations. Another bit of history was that in 1744, Anson returned after a 4-year circumnavigation. And interestingly, there were only 4 men killed in naval battles out of his fleet, but over 1,000, or fully a third of his sailors, had died from scurvy. And not long after, the Scottish naval surgeon James Lind was put on assignment, and on his boat, by careful documentation, 10% of his men a year later had come down with scurvy. And unlike the recommendation of the time, Lind did not use bloodletting. But what he did do, apparently based on the earlier anecdotes, which you would have thought would have been somewhat definitive, was he took 12 of these sailors, and as he says, their cases were as similar as I could have them. He describes them. They lay together in one place and had one diet common to all. So if you will, that's eligibility in Table 1. Then he goes on with the treatments. 2 of these were ordered cider, 2 others elixir of vitriol. I don't know what that is, but it doesn't sound good. Vinegar 3 times a day, and also had their gruels and other food. I mean, if you're eating gruel, maybe it's okay to put vinegar in it. But then the worst of the patients, of course, had other things tried. And then 2 others had each 2 oranges and 1 lemon given to them every day. And they continued 6 days under the course. Results. The most sudden and visible effects were perceived from the use of oranges and lemons, one of those being fit for duty at the end of 6 days. So here it is, an experiment by any measure, a true experiment founded on the principles from Anson and earlier history. But he dropped the ball, in a sense, didn't publish this for 6 years. And then, believing the result appropriately, he was trying to figure out how to get fruit, the magic bullet, at sea. And so he developed a concentrated version of lemon juice. And that required heating and evaporation, which, as we now know, destroys vitamin C. And so when that concentrate was taken to sea and tried, it wasn't effective. And so that reflected badly on the original experience. But the work did attract the attention of this physician, Gilbert Blaine, who incidentally nickname was Chilblain. And he was preparing for a long sea voyage and posting, and also monitored mortality rates and found this huge number, 1,500 of his 12,000 sailors died from scurvy. And then they did supply the fresh fruit, hence the name limeys for British sailors, and the death rate was cut in half. But Blaine's work was not as tightly controlled as was Lind's. And only when Blaine became very influential on the so-called Sick and Hurt Board, which oversaw the health of sailors in the Navy, 15 years later, so the clock is really running here, was he able to really implement scurvy control. 50 years it took, with this convoluted history and insights, mistakes, and so on, for the British to adopt effective scurvy control. But they were way ahead of the time. Other nations weren't so quick. And interestingly, you might claim that James Lind saved the British Empire because in 1805, during the Battle of Trafalgar, the British fleet was able to keep the French out of Great Britain. And they did so in part because the sailors were healthy and didn't get scurvy. Napoleon's army and ships were much stronger. So you might argue that this is key in the history of clinical trials, and the insight saved the British Empire. Other experiences that really edged into the field of designed experimentation. An example from the early 1800s, the military surgeon Alexander Hamilton. He was a skeptic with regard to bloodletting, and his experiment was to take over 300 soldiers and divide them into 3 groups. 2 of the groups were treated without bloodletting, one by the conventional means. And you can see here his version, if you will, of unbiased treatment allocation. The soldiers were admitted alternately in such a manner that each of us had 1/3 of the whole. This is actually not a bad idea. The only reason we use randomization formally is because we want to control the impartial allocation as opposed to leaving it to the order in which individuals came. Same care and accommodated with the same comforts. And then you can see the mortality count there. So the conclusion was that bloodletting was ineffective for this variety of conditions, if you will, a heterogeneous study cohort. But bloodletting was going to be around for a long time. Hamilton, like Lind, didn't actually, at least Lind published his results 6 years later. Hamilton did not. And his work was actually discovered 150 years later, they say, hidden in a trunk at the Royal College of Physicians of Edinburgh. Interesting if it's true, and there's some skepticism around this point as to whether this work was truly rediscovered, or some questions were even raised about fabrication. But it's probably genuine, and a tragedy that it was not published and disseminated early. So important threads picked up by the mere observation and, if you will, flirting with, or in the case of Lind, truly designing an experiment. A little bit about the role of chance then and how it's influenced our ideas. And they say that a picture is worth 1,000 words, and I hope in this audience I don't need to do more than just show you this picture and understand immediately the important role of chance in the developing of valid and powerful therapeutics. This also, although was an important story, certainly in my formative years, maybe is receding into the bowels of history. If you don't know what I mean by this, ask a colleague. Another business of chance, the surgeon Ambrose Paré, again, now I've backed up in history a little bit, and this drawing here is apropos. It's an unknown German artist describing wound care. These wound men, if you will, were very popular guides throughout history for physicians treating these kinds of injuries. And you can read about this in the book there. But it was a custom of the time to treat battlefield injuries with boiling oil to cauterize them. And Perret just ran out of oil. So he applied whatever else was at hand, which happened to be eggs and oil of roses and turpentine. Turpentine can be fairly noxious, but has had various therapeutic uses throughout history, including recently. And then here's what he observed. I raised myself early to visit them. When beyond my hope I found those to whom I applied the eggs and turpentine, feeling but little pain. They had slept through the night. The others to whom I had applied boiling oil were feverish with much pain and swelling about their wounds. It was clear to him instantly with this experience that he should not be using boiling oil ever again. Interestingly, he was the official royal surgeon to 4 French kings during the 1500s. But he was a believer in bloodletting. So here again is this paradoxical embodiment of insight, but not really appreciating the natural experiment that he participated in. And here's what he said about leeches. You know, he was very sophisticated. You can actually make a leech angry if you handle it too much, but then you can calm it down if you treat it right. And so he used bloodletting extensively throughout the royal court of the French kings. Jenner's work relied, in a sense, heavily on sophisticated observation and chance. The milkmaids who got cowpox never got smallpox. And so Jenner figured out, in a way that's well known and almost obvious to us now, that he should give cowpox to people as a preventive for smallpox. And then he tried to give smallpox to people who had cowpox, and that didn't work, demonstrating the efficacy of the treatment. I'm going to move on then and talk about comparisons, direct comparisons, and appreciation of bias. This audience may be familiar now with this picture. I've used it several times, as have others. But you're all epidemiologists. When you look at this picture, what does it say to you? Smoking's not good for you, right? I mean, we take immediately our milieu, our cultural awareness, our science of the day, and we interpret the picture. And one could argue, as I'm sure Kurt would, because he loves to argue, that this would in fact be an interpretation, the interpretation. But if I go on and tell you that this was a Van Gogh, and that it was painted long before the effects of smoking were known or appreciated, then you have to interpret the picture historically in a very different way. And one of the ways you might interpret it, and this insight came from Kara, who I mentioned earlier, was that at the time that Van Gogh was experimenting, there was a tradition of in Dutch still lifes of putting the quill in the bowl of fruit. You've probably seen that somewhere in your wanderings through the museums. And that this cigarette stuck in the skeleton, which may have been a study of form and shape and structure, could easily have been the quill, if you will, in the still life. But it had absolutely nothing to do with the evils of tobacco, which were unknown at the time. In fact, it was probably thought to be rather healthy. So we filter these ideas by what we think we know, either forward or backward in time. Now, we could argue that the modern era of counting and assessing scientific evidence began around the time of the mid-1800s. One of the figures there is Louis, and he was also a challenger of bloodletting. and there's a reference to his treatise on the subject. He directly compared large numbers of patients treated in different ways, and thought I would show you this picture here, which came from, I have to consult my notes. This was a tuberculosis congress held in Paris, and this is a picture was taken at the gravesite of Charles Alexander Louis, who died in 1872. And interestingly, at the right of the first group is William Osler. So I thought that was an interesting footnote to show you here. Anyway, Louis recognized that you could overcome this difficulty of lack of comparability with a large enough sample. And here's a quote from his work. You can mutually compensate, if you will, and the differences can be disregarded without sensibly affecting the exactness of the results. So again, a very sophisticated notion. And through his work and this method, he concluded that bloodletting was really not as effective. Interestingly, this numerical method was resisted, and in fact, the numerical method, statistics, have been resisted by every basic and clinical scientist from this very first articulation even through the modern era. And I'll say a little more about this in my afternoon talk over in the Cancer Center. But the notion is that the average experience is somehow not relevant to the individual patient. Now, if that doesn't sound like personalized or precision medicine, I don't know what does. But here it is 150 or 170 years later, and we're still hearing echoes of the same irrelevance. But the counter-argument was that you couldn't really know about safe and effective therapies unless you had treated an adequate number of subjects. And he said, without the aid of statistics, nothing like real medicine is possible. Debate continues. On the empiric side, we have a figure like Paracelsus, who I mentioned Celsus earlier. His desire was to be beside Celsus, if you will. This was the itinerant physician, Philippe Theophrastus Bombastus von Hohenheim. And it is said, although I'm not sure it's correct, that the term bombastic came from his middle name. But he was a very direct opposition to authority of the time. He argued for the empirical use and observation, and therefore sometimes credited with founding the science of pharmacology. He refused to teach in Latin, thinking that it was obscure, and but yet, in spite of these kind of earthy and forward-thinking insights, he carried with him always a belief in astrology and the occult, and interleaved these ideas into his more sophisticated one. There's a wonderful biography of him for those of you who are interested in reading more, called The Devil's Doctor. And this quote, how he proclaimed the absolute authority of experience, argues against the theories of the ancient, but then, like a lunatic, seems to converse with demons, fasting in the morning, drunk in the evening, presenting exactly every idea in the order in which it came to his mind. Interesting guy. Thomas Percival, I mentioned earlier, as the originator of the notion of medical ethics in the early 1800s. He had an interesting series of polemic essays, which I believe you can find right here in the historic library at Welch, about the rationalism or dogmatism, if you will. Dogmatism became rationalism during the Enlightenment. Theory is absurd and fallacious, always useless, and often in the highest degree pernicious. The annals of medicine afford the most striking proof that it hath in all ages been the bane and disgrace of the healing art. And yet, in the next essay, the rationalist enlarges the boundaries of science in general and adds new discoveries to the art of medicine. The rationalist has every advantage which the empiric can boast from reading, observation, and practice, accompanied with superior knowledge, understanding, and judgment. So, in a way, he's laying out exactly the dialectic between empiricism and rationalism and why we need both of these. We need the biological theory that embodies itself as rationalism and in a way makes predictions and helps guide us as to where the errors are. But we need the empirical side. We need to test the treatments and make direct observation, because only in that way are we going to discover where we might have gone astray. Bernard, the great physiologist, was an advocate for the experimental method, but interestingly despised the statistical method. We know now pretty much that you can't do real experiments without an underpinning of statistics. He was an anti-empiricist, if you will, sort of the rationalist or dogmatist. Medicine is destined to escape empiricism little by little, and will escape in the same way as all the other sciences, by the experimental method. And yet, interestingly, he denigrated this growing field of the numerical method and statistics, that area that is applicable everywhere in science. Yet he didn't recognize its utility in his own. So, drawing near the end of the time, I want to leave some time for discussion and questions, but I'll deal with just one more issue that has to do with masking, since it's a very important principle in our methods, and then I think I'll just stop short, Taddy, if that's okay, and take some questions. Interesting chapter in the history of masking comes from this fellow Franz Mesmer. You've heard of mesmerization or hypnotism. He was a promoter of the beneficial effects of magnetized water. Maybe not so different from some of the health claims that are made today. And Louis XVI set up a royal commission to investigate this because it was becoming quite an issue of the time, and Benjamin Franklin was among those who were lined up to test these claims. And so there was an experiment, even though this was pre-1800, formal experiment, if you will, 5 identical glasses of water, one of which had been magnetized, and then the subjects of the experiment, volunteers, if you will, randomly picked one and drank it. One patient fainted, consistent with the effects of magnetized water, but it turned out she had drunk regular water. I don't know if that's the placebo effect. And the Royal Commission concluded, after all these experiments were done, that magnetized water was the same as plain water. Here again, they had right at their fingertips this notion of the placebo effect, if you will, but they didn't make the leap to other areas of medicine. Now, you might laugh at this and say, well, magnetized water, anybody would have known what a ruse that was. But if you read the history of homeopathy, you will not laugh at the notion of magnetized water, and that is a persistent concept that water can be induced to have memory right up to this very moment. So we're not so much smarter as we think. Haygarth, then, who's often credited sort of with the first formal use of masking, was charged with investigating these tractors or metallic rods that were used to treat arthritic conditions. And he did so in what can now be viewed as a pretty sophisticated way, using double masking. This isn't much longer afterwards. Prepare, the language is a bit funky. Prepare a pair of faults exactly to resemble the true tractors. Let the secret be kept inviolable, not only from the patient, but from every other person. Blind leading the blind. Let the efficacy both be impartially tried. Cases should be accurately stated, fully given in the words of the patient. Wooden tractors were employed. All the 5 patients except one, again, interesting language, assured us that their pain was relieved. All were in some measure relieved, and by a second application, and so on. But in fact, the tractors were fake. So the demonstration that with the masking and removal of the ascertainment bias, one could render a much more sophisticated test. So I'm near the end of my appointed time. I have more to say about randomization and some other things, but again, this subject is extremely long and deep, and I think it's best at this point just to see if I've stimulated your comments or questions, and we'll pick up the remainder of this maybe another time. Thank you very much. Yes, sir.
Fascinating way of looking at the historical statement going way back when, and it makes me wonder, is it really true that people at any point would not have understood fair comparison? What is it that they don't understand? And I guess one way of, for me, understanding what they don't understand is to look at how today unsophisticated people will rely on, this is a doctor and he gave 95 water and these things got better. For all the junk science that's out there and books and so on, for somebody that's making a point. And I think the lack of understanding there is that people don't, I see it more as not understanding confounding than that comparison is useful. Because I think, so when somebody says, I gave you this treatment, you got better, they don't realize that you may have gotten better for another reason. They don't realize that there's a possibility that there's something mixed with the effect of the treatment that caused him to get better. Going back to the time of Daniel, people presented with 10 people getting lemon juice and 10 people who didn't, and all the people with lemon juice got better, would be persuaded.
Well, I can't really disagree with your interpretation, but I'll say 2 things. One, you sound just like a Hopkins professor. And number 2, you may be giving people more credit in terms of sophistication. There's a lot of respect for authority historically where people will disbelieve their own eyes and ears and hands. And if you want a few examples of that, come to my talk this afternoon, where I will argue in a very perhaps indirect way, but definitive way, that we are not much smarter today than these examples that I'm showing you historically. Astonishingly, but the evidence suggests that we are not a heck of a lot smarter. And I don't know, obviously we know some things that were unknown, but the thought process, which I think is what you're asking about, that we use to approach even our foundational questions in basic science, and particularly those ideas leading into the therapeutic pipeline, are somehow defective, seriously defective, leading us to make a high fraction of mistakes. And that we have to examine those thought processes that it goes beyond the authoritarianism and lack of appreciation of confounding and so on. I think they're more rudimentary errors. But I don't disagree with you. At some level, there's kind of a sophistication where, yes, I understand the idea of comparison and I might be convinced by that, but there's a subtlety of confounding that I don't appreciate. But you can get very smart people to disbelieve their own eyes and ears under the right circumstances, which I'll argue we commonly encounter. Kurt.
Steve, what are you, an empiricist or a dogmatist?
Oh, I'm neither. Yeah, but that's only because it's all you understand is dogmatism. No, I'm just kidding. No, I'm neither, and I can tell you honestly, I've tried very hard to achieve the right balance, and I think that clinical trials, just to come to the theme here, are really, really an important tool in this proper balancing of dogmatism and empiricism. And there are cases where we take the methodology of trials and we apply them purely empirically. And I don't think that's science. And I argue with my class every year because we talk about, can I do a scientific test of homeopathy? Can I do a scientific test of therapeutic touch? Can I do a test of intercessory prayer? And invariably, year after year, the students say, yeah, you can do that. And I say, no, you can't do that. You don't understand what's actually going on. So I think that the balance has to be there. Otherwise, it's not actually science. Dr. Coffey.
Well, one of the problems is the human nature, and everyone in this room, many people speak the famous SAT test, where you put a sentence up. It has 6 SAT sentences, 3 of them are up. And you give that to over 1,000 people. I have actually done it. 80% of them get it wrong because they don't believe arbitrary points in their mind. They just count the F's and a lot of that. And so what does this mean? It means our mind is trained as we grow up to see what we think is important. And so that carries over into medicine all the time. We only see what we believe to be correct, whether more than temperature for gold or the baby in the sun. We believe that.
You just gave my afternoon talk. Thank you.
Okay. But how do we overcome that with statistics? That's my first question. And my second question is, I was looking at some trials one time involving alpha blockers for BPH older men, and we didn't want them to get into night and fall. And so the 2 alpha blockers, one in the United States and one in Japan, had nobody breaking their hips in these trials. And that was way below the possibility of thousands of numbers and falling at that age. Turned out the protocol exclusion. They didn't put any patients in that ever had any vertigo or fell or anything. And so by taking those out, they had a very low fracture of the hip thing. But when they put it into the public trial, now things have had a lot. Here come all the lawsuits, all the lawyers we ever had. And how do you correct for those 2 things? One is the obvious bias that you can't get out. 2, protocol exclusion.
Well, the second one might be a little easier to deal with first. It's a classic phenomenon that I'm sure everyone in this room is familiar with, the healthier status of volunteers on clinical trials and the fact that we see generally lower mortality rates and lower complication rates and so on. I'll quote Meinert, and even though he doesn't like me to quote him, he says the easiest way to cure disease is to do a trial. And, right, Kurt? Is that close enough? Yeah. You said it more than once. Anyway, we recognize this effect. So the solution to the riddle, I think, comes down to what is it that we're trying to learn in that trial? Are we looking for the biological effect of alpha blockers? And if we are, and if falls and other issues like that are a mere nuisance with respect to the biological effect, then it doesn't matter that the cohort is healthier and less at risk of falls, because the comparison of the 2 alpha blockers will come forward and will be biologically valid. If, on the other hand, we're interested in the effects of the therapy applied to the broad population, then clearly a restricted eligibility criterion is not going to serve that purpose. So we're led into what is stereotypically called the difference between an efficacy or a biological efficacy objective versus an effectiveness objective, the latter being what happens more broadly. But we do see this time and time again, and I don't think, given the nature of our ethics overlay and the requirement for self-selection of study participants, that we're going to escape the fact that generally healthier people participate in trials. They are generally healthier, they're generally more educated, they're generally whiter and less minority, and so on. They generally have higher functional status. And it's very, very difficult to overcome that. With regard to the other issue about what we can do about these perceptual biases, there are some things, certainly in the design of an experiment, with the protocol and requirement for certain kinds of observation that would at least partially, if not completely, compensate for the perceptual weaknesses that are hardwired into our brain. For example, if we said, well, I don't want you to read that slide with all those F's on it. I want you to take an electronic version of that slide and send it through a computer program that counts the F's. So by instrumentalizing the perception, if you will, we can be pretty darn sure how many F's are there. So we remove it from the subjectivity and the perceptual biases. Now, we can't always literally do that kind of thing, but we can do things like that per protocol and using other methods that will help correct it. Alvaro, yeah.
I think that it's easier to be one of the others. It's easier to be a dogmatist because you don't have to work. You only have to do things. It's easier to do an empiricism because you don't have to do it. But how to combine the two? How to make the two? Because you told us either are non-scientific. How to combine the two?
Well, I think that's our challenge, and I can't give you the lessons for exactly how that's done, but I think that's what we should be teaching. I think that's what we should be practicing. I think we shouldn't be afraid to say that either extreme is non-scientific. We don't want to take our medical advice from Galen because he said that was the way it should be, nor do we want to take it from any authority figure. We need to take the suggestion for what might be important questions, what might be key to our biological knowledge, and we need to test those in fair comparisons. I think that works, although I can't tell you exactly on an individual basis how to do it. It certainly works in this domain of evaluating therapies, therapeutic comparisons, because my personal belief is that properly done clinical trials are, in fact, this ideal balance. It's only the improperly one, so to speak, which we're all familiar enough now with methodology that we can generally tell the good methods from the bad. I think that where this notion may not have penetrated adequately is in the more foundational sciences and basic sciences, where there's still a lot of theory being accepted, but also a lot of theory necessary. When the experiments are small, for example, how do we get from a little bit of evidence to the next step in the investigation? And we need theory to bridge that gap, but we need validated theory. And that's why we can't do a scientific test of things that are belief-based. Because once they're belief-based, we believe some things even without evidence. And I could step on everyone's toes in the room, but I won't do that. But we all do it. Maybe later, Kurt. We all have those things. The core of our beliefs are non-rational. Why do we know that the scientific method works? You can't really prove it. we believe that. But once we take that belief and use it, then we can probably find the truth and reinforce the belief. It's not an easy problem, but so Alvaro, I can't answer the question, but I think that we must strive for the appropriate balance. Taddy.
I think part of our problem is understanding that it's very right now. And there's been this tendency that, here's what I take, here are the results, and there's no thinking. So, we might just analyze a couple of variables or, small possible amount of something else, something else, reporting of results, and we don't rely on authors to also think. And so I think we have moved away from telling a story and things where actually you're the one doing the trial or doing the perspective or whatever, or anybody else about that, whether it's the literature and their own data. And so I don't think we as teachers are doing a very good job with our students in terms of paying one paper.
Yeah, I don't disagree at all. And, the pressures of the times, the pressures of space and word limits and so on, among others, I think contribute to that. Dr. Coffey.
This is a little heresy, but I'll throw it out since we said we could step on it. I feel in my heart, I don't know this, that there's something wrong with our epidemiological approach in studies. And that is that we've known for 50, 60 years that there's big differences between prostate cancer and breast cancer in China, and when it moves to our culture, it goes up, and then the next time it goes up pretty high. Now that's such a cutting, dry observation that you would think after half a century we'd have some idea what that factor is. I have no idea what that factor is. And I begin to wonder if there's something like an emergent property or something I don't know that, there's something going on here that our mathematical approach of linear changes may not be inappropriate to answer the question. Are we in real trouble with the mathematical analysis of epidemiological?
Well, I don't think so, honestly. I mean, it's glib, but I could say that maybe the explanation for your observation is reporting bias. And I know there's a little evidence that prostate cancer, at least in Japan, may have been underreported.
The reason I say that is the answer that we get. But I spent with Wu Xingpian, the head of the college of medicine one time in China. He trained with Charles Hutton. And so we sent pathologists over and compared slides over his data and all this. He was well trained in prostate. He said he saw more prostate cancer in the United States in 1 week that he saw when he was head of Beijing and working in the capital in a year. And he said it's just fundamental. He said, but all the Americans would say it's underreported. And then he showed me the data they had. Showed it wasn't. And so I just want to throw that in. It made me feel that we only look, we talk about it, but phase shifts, like water to ice and things, it's very hard to analyze in these types of things, particularly emergent phase shifts.
Well, you have confidence in the adequacy of the reporting based on that experience. I personally, and I'm sure others in this room even more so, have confidence in the adequacy of the analytic methods and population models that are used. So I would argue that we need to look elsewhere for the solution. But I don't want to come to this department and defend it. They can do it themselves, Taddy or Kurt, Alvaro, anybody. So, okay, well, thank you very much.